photoreceptors and treated them with a visual chromophore and exposed them to light and induced cell death. Using that model, we then treated them with a Fas ligand blocker, and you can see here with light we induced a considerable amount of cell death, and that was, retina specialists always travel with their own laser, was significantly attenuated with Fas antibody, but not nearly as much as if you inhibited with caspases. But the interesting thing we learned from this experiment was this. If one looked at necrosis instead of apoptosis, it was sort of, at least paradoxically for us, increased. And so here's the anti-Fas antibody, but the anti-caspase antibodies, and then combined anti-caspase with anti-Fas, saw a big increase in necrosis. So we weren't diminishing cell death. It was just sort of like a balloon. We squeezed one end and the other end got bigger. And so our sort of take-home message from this set of experiments was that, at least in our phototoxicity model, that there were multiple divergent and overlapping pathways with some redundancy affecting cell death, and that it would be unlikely for us to sort of find, and my background is in genetics, a genetic smoking gun that sort of figured out this entire process. But what we were left with was a cell-based model of retinal degeneration, again with these 661W cells that we treated with visual chromophore, and we could predictably turn on the light and in 4 to 6 hours produce 80% cell death. And so what we decided, rather than pursuing genetic solutions to this issue, was to perform a chemical screen. And again, that was because of this multifactorial cell death that we were seeing, involving both apoptosis here as well as necrosis. This is dark and this is light. And so what we set out to do was screen a chemical library to find chemical compounds that might affect both cell death pathways. And that's because, as many of you know, drugs are quite promiscuous and dirty and can affect multiple pathways. And so the library that we chose was the Prestwick library, which is composed of FDA-approved compounds. It's 1,200 compounds. And the reason we chose this library is because these compounds have been around for a while, and lots of their mechanisms and pathways that they affect are known. And so when we did the screen, the first question we asked was, well, are we seeing anything that's biologically plausible? And for genetics, when we do screens, we have very sort of set criteria in terms of false positives. And so for us, in screening 1,200 compounds, it's very easy to stumble across things that aren't real signals but purely false positives. And so we did what would be analogous to an ingenuity analysis, where we looked at the different pathways that the compounds affected, and somewhat reassuringly to us, we found that the biggest group, as you would anticipate based on the talks and things we've learned today, were calcium and ion channel blockers. Another big category was anti-inflammation with compounds like minocycline, and the category we're interested in were these CYP450 inhibitors, because these were the top 20 compounds, with sulfophenazole being the top compound, almost 3 standard deviations beyond the mean for the screen. And sulfophenazole is a cytochrome P450 inhibitor. And cytochrome P450s have some history in terms of retinal degeneration. Cytochrome P450 inhibitors, for instance, is implicated in Beatty's corneal retinal dystrophy. And then some isoforms 46A1 and 27A1 are associated with age-related macular degeneration. So again, the concern was, well, is this compound a false positive? And so the next step we took was just simply to bring it back to the laboratory. The high-throughput screen was performed on 384 well plates in a roboticized facility. It was to take it back to the lab and re-examine it. And in my experience, anytime you can do something in science and not need a statistical analysis or p-value, it's usually insightful. And so when we looked under the microscope, this is what we saw for sulfophenazole. Here's the dark control, and you can see a significant percentage of the cells maintain the spindeloid neuronal configuration. And these were some of the other compounds in our hits. And again, minocycline's been studied for many years. Mark Cho has used it in a similar assay. And so when we quantified it, you can see sulfophenazole did indeed offer a neuroprotective effect for the cones. And so we did a dose response curve, thinking if a little of something is good, more of something may be better. And indeed, when we increased the concentration of sulfophenazole, it did offer more protection. The next question, though, was, well, if it is in fact neuroprotective, how is it having that effect? And as I mentioned to you earlier, in our assay, we saw cell death both by necrosis as well as apoptosis. And the concern was we were going to see this sort of push-pull again with sulfophenazole perhaps decreasing apoptosis while increasing necrosis. Interestingly, when we looked at sulfophenazole's effect on apoptosis, it was decreased as well as decreasing necrosis simultaneously. It did this in part by stabilizing the membrane potential that we see here, similar to the dark control, but also by impairing caspases. This is flow cytometry, and you can see the configuration of light plus sulfa maintains something similar to the dark morphology. So sulfophenazole is a specific cytochrome P450 2C9 inhibitor. And the question was, well, if we're seeing this neuroprotective effect and it's inhibiting apoptosis and necrosis, through what mechanism is that? And a good candidate would be through CYP2C9. And so you can see that CYP2C9 is expressed in our cell line and that its expression is increased with light exposure. Next question was, if in fact it was mediating its effect through 2C9, we should be able to provide some photoreceptor rescue by 2C9 knockdown. And so we performed RNA interference and did see some protection with 2C9 sRNA. Again, this is a murine photoreceptor cell line, so this is in a typo. 2C29 is the murine homologue. Finally, the question was, if 2C9 is mediating the effect, what's the mechanism? Well, what's known about light exposure is that it stimulates phospholipase A2. Phospholipase A2 does many things, but one of them is generate arachidonic acid, which is a substrate for 2C9. 2C9 metabolizes arachidonic acid into these EETs, which is short for epoxy-eicosapentaenoic acid. They're simply eicosanoids that mediate signaling, in particular calcium signaling. And so you can see these isoforms in our cell line increase calcium flux. The calcium flux, if this is the potential mechanism that sulfophenazole is having its effect, should be attenuated. And here you see calcium on the x-axis, stimulated by light, and again attenuated by sulfophenazole. AACOCF3 is a phospholipase inhibitor and again provides some protective effect. So in summary, we performed a chemical screen. We identified a novel cytochrome P450 inhibitor that may be having its effect through a specific isoform, 2C9. Obviously, all of this was done in a cell-based model, though, and the proof in the pudding will really be is if we can extend this to a more complex mammalian system. Fortunately, the light-induced rat retinal degeneration model has been around for 40 years, and it should be relatively easy to test that. There's also a mouse cytochrome P450 2C knockout that exists. Ching is responsible for the great majority of this work that was presented. From the lab. He'll present this at ARVO this year, and Joe is an MSTP student in the lab. Chris Palczewski has been very helpful, both in terms of suggesting potential experiments as well as resources and time. And again, many thanks to Hope for Vision and to you for your time and attention.
Good afternoon. Today I'm going to talk about DHDDS and retinal degeneration. So DHDDS K24E genotype was discovered in Miami through whole exome sequencing of a family. And from the presentation that was done by Zuckner, a subsequent group then found that DHDDS K24E accounted for 12% to 15% of autosomal recessive RP in the Ashkenazi Jewish population. DHDDS is highly conserved. If you look at this diagram, this is the DHDDS sequence, and you can see that the 24 position is highly conserved, and the bluish area is basically the active site. This is a 3D anatomical model of the DHDDS, and this is a wild type to the left, and this is the K42E. So the basic residue R38 and R47 here, they are part of the pocket that holds the FPP. And what happens when you have a change in K24E is that the basic residue of lysine at this position is changed to an acidic residue to glutamic acid, which then pulls this R38 position away. You have a looser pocket, and this compromises the binding of the FPP. This shows the zebrafish histology, and this is the morpholino oligonucleotide designed against exon 1 and intron 1 of the zebrafish DHDDS gene. It was injected to fertilize eggs. And on the left, these are the controls, and these are the injected fish. And basically what you see in histology is that in the normal you have photoreceptor outer segments, and here they're either absent or they're just a small part of it is present. And on the bottom are the PNA staining of the outer segments, showing that it's present in the wild type and it's basically absent in the treated fish. This is a DNA construct for the knockout mouse for DHDDS. And in A is the construct, and it shows that this creates a fringe of the DHDDS, which then produces an DHDDS null allele. The flipase then removed the region flanked by the two FRTs sites to create the DHDDX flux, which is shown in B. And the flux exon 4 can then be removed by Cray, and which is shown in C, and that creates a conditional knockout of DHDDS. This shows the DHDDS knockout mouse, and basically knockout mouse, they don't survive. It's a lethal mutation, and this is the E7 embryos, and about 75% of the embryos developed well at E7, and the other 25% fails to develop, and basically this is a lethal mutation when you knock out DHDDS. On the other hand, when you have the heterozygous mouse, here are the retinas of the DHDDS heterozygous mouse. And on the A to the left, you have a 1-year-old heterozygous DHDDS mouse. And when you look at the retina, it's pretty much normal. It has a normal morphology. And then to the right is the age-matched wild-type mouse, which, again, they're very similar. This shows a conditional knockout of DHDDS in photoreceptors by AAV Cray. So these mouse in the right eye, which is shown here in A, and their left eye is shown in B. So in the right eye, they're injected subretinally with AAV, short rhodopsin mutation with GFP. And in the left eye, they're injected with the AAV short rhodopsin promoter with Cray. And you can see that when you have the conditional knockout this way, that the retina is definitely affected. This is basically the right eye, the normal control. On the right, the one that's injected with Cray, you have really decreased photoreceptors here. This is the normal. And also the outer nuclear layer is really thin. So basically, the animal model shows the retinal degeneration. This is the conditional knockout of DHDDS in photoreceptors genetically by long-mouse absent promoter Cray. And on the top, it's the Cray control, which is normal, and on the bottom is the knockout and the conditional knockout. And you can see again that basically the conditional knockout produces retinal degeneration. There is loss of photoreceptor outer segment, and also the outer nuclear layer is thinned. So in conclusion, DHDDS is required for photoreceptor survival. The function of DHDDS cannot be compensated by other genes. In photoreceptors, dolicho cannot be supplied by other cells in the retina. They have to be synthesized in situ. And to further study the degeneration, we need to make a K42E DHDDS mouse by the knock-in technology, which is underway. So I think the upshot of all this is that the conditional knockout mouse shows the retinal degeneration, and it's going to be a very good model to test the different treatments. I want to thank all the funding sources, and particularly NIH and also Hope for Vision, and also our other funding sources. Thank you.
Okay, I'm going to talk about why mutation DHDDS kills photoreceptors, not other cells, because DHDDS is an enzyme you can consider as a housekeeping enzyme that everybody needs, every cell needs it. If you, as Byron just mentioned, if you knock it out, basically you cannot survive. So this mutation causes retinitis pigmentosa. It's a key enzyme to make dolicho, which is obscure name I learned several times before I can memorize it. The mutation weakens the binding, and therefore you probably get dolicho deficiency. And the big question is, dolicho deficiency is all over the place. Why photoreceptors are so susceptible to dolicho deficiency? The functions of dolicos are probably the key. That's bothering us for quite a long time. We're still thinking about why photoreceptors are so vulnerable to this problem. So let me give you some kind of background of what dolicho is all about. Dolicho means that's long. So it's a long stretch of one of these things called isoprene. It has many, up to 24 units long. And it's basically a long chain lipid. This is 3D. I pulled out from literature. 3D of image of dolicho. It has a central core area and two arms around it. And how it's made? It's made from FPP adding number of IPP, the FPP, IPP. Who is doing it? DHDDS. So if you knock out this, you don't have this, which is, I don't know if that's okay, but if you don't have this, you're in big trouble. So that's because dolichophosphate is a lipid carrier for glycosylation. So how it's dolicho is in the membrane? It's that's 3D image of dolicho buried in the membrane. We don't know what it does. A lot of suggestions we play with. This is dolichophosphate. So if you look at this, it's vertical. It's parallel to this panel. And dolichophosphate is rather stick ahead out because it needs to get the sugar chain. So this is how the sugar chain was made. You get the dolicho and the phosphorylated, and then from the dolichophosphate you grow this chain, and the long chain flip over to the other side, and then eventually transferred to protein. And then the phosphorylation is going to get trained, add more different sugar, and eventually get secreted out or stay in the cell. So that's originally the two papers published all talked about glycosylation. So we thought, well, must be it. Glycosylation is not new. We know there's kind of disease called CDG, congenital disorder of glycosylation. That's well known. And the best test on that is to test the serum transferring this IEF pattern. So those are from the family from family members in Miami. This is a negative control. This is a positive control. So this one has glycosylation deficiency. The other one has not. I mean, here you got patient 1, 2, 3, 1 carrier, 1 wild type. You don't see differences. So that's one thing we don't think that is glycosylation problem. The other thing is that when you get this disease, it affects everybody in the body. But in our patient, there's only one thing. Oh God. Okay. So then we think about what dolichol does in the membrane. Dolichol, when you add to the membrane, it increases the fluidity and it increases the transition from liquid crystal to this H2 phase. So what does it do? Because it's in the membrane. This is an outer segments of photoreceptors. That's the image. And outer segment has all of these discs. This disc is a pancake with very sharp turn here and a lot of rhodopsins decorated around. And what does it do? Okay, and the other thing in the photoreceptor is that you got this UCLA marching band. That's nicknamed UCLA marching band goes in 10 days, it's gone, right? So it's gone where? It's gone to RPE cells. So basically, a rod needs to make 10% of the outer segment every day, and it's gone. You have to make it anew. Cannot recycle. It has to make this much membrane area, and it's equivalent to twice as much as the cytoplasm, the envelope of the outer segments. So if you go back at this, its effect, it promotes visual vesicle fusions. That's Deborah mentioned about. And if you affect vesicle fusions, then you have trouble getting lipid out to the outer segments. Then outer segment renewal is impaired. Okay, we think that's one of the mechanisms. The second mechanism is that you have these sharp turns, and it generates a lot of tension in the lipid membrane, and the lipid dolichol serves as a lubricant. In this, it reduces all the tensions, so you can bend this membrane sharp turn, no problem. And when you decorate this thing, you have a dimp all of the membrane, and that generates tensions as well. So if you have dolichols, you don't have that problems anymore. So if you don't have enough dolichols, then you could have a problem. The third hypothesis is that actually receptors, like photoreceptors, like rhodopsins, like to sit in this H2 phases rather than this what do you call it? This liquid order microdomain. And if you don't have enough dolichos, you could affect the phototransduction itself. So we think that's the conclusion, that it's very unlikely that the problem caused by the DHDDS mutation is glycosylation. Rather, we think it's dolicho deficiency, the free dolicho deficiency, rather than the glycosylation deficiency. And the membrane metabolism, unique membrane metabolism of photoreceptor outer segment, that is the key to make them vulnerable to dolicho deficiency. So the future direction, what we need to do is to get a mouse model like Byron already mentioned, and then we have to use this model to recreate the disease and recreate this in the retina to test the hypothesis, and the model will be used also, of course, to evaluate potential treatment. We have several in our pipeline. And finally, that's where we got money to support this, especially thanks to Hopeful Vision. Thank you very much.
Well, I just wanted to say thank you. Is this on? Can you hear? Thank you, Betty, for the wonderful invitation. It's really very exciting to come back, and I feel very nostalgic about coming back to Miami after 5 years. And I also want to say that what a beautiful story about the DHDDS, do I have that right, gene, and how quickly that has evolved. You must be very excited to have worked on that. Congratulations. Okay, well, I'm going to talk about something that I started in Miami, and it's about a novel G-protein coupled receptor, an orphan, that we're getting some exciting results on. So I started this work with Steve Schwartz, who's a retina specialist, and Steve was working in the clinic and starting to use corticosteroid drugs, and was running up against an old problem that had kind of gone away, but now the corticosteroids, when they came back, it arose again, and that was the problem of steroid-induced ocular hypertension, or glaucoma. And he was mostly using intravitreal triamcinolone at that time. This was about 4 or 5 years ago. And he was getting, in many patients, a response that was very high intraocular pressure, in some cases intractable, even requiring surgery. And so Steve wanted to know, what is the genetic basis? Why are some of these patients more susceptible to getting this very high spikes of intraocular pressure and rather than others? And so we wanted to elucidate the genetics. And he was interested in ultimately a prognostic test that would make steroids safer if you could identify the mutations that were predictive. But he convinced me to work with him on this because I was very excited about the idea of this being an unbiased screen that would reveal new pathways for ocular hypertension. So we went ahead and worked with Scripps Florida, which was new at that time, and did a screen on the old 500K microarray chips. They're much bigger now. And we had a pool of 52 patients. It turns out, doing the statistics, that that is actually enough for a small screen because that's powered high enough if we do a continuous variable analysis, because the pharmacogenetic effects can be so large. And it turned out that we did get some results in a few genes that came up at very high p-values that were past the bar that geneticists would accept. So first, let me just back up and tell you a little bit about glaucoma, since we've been hearing mostly here about retina defects. So glaucoma is all different sorts of diseases caused by different things, but it has in common this visual field defects and also this unusual optic nerve cupping that's this very distinctive shape. And it's caused by problems in the outflow of the aqueous humor in a lot of cases, probably still the major risk factor. And so it's not that the aqueous humor, it's not that too much aqueous humor is made, but that it doesn't drain out through the outflow pathways properly. So this just shows you a picture of what those pathways look like. We have a freeze fracture showing the spongy nature of the tissue, And then this schematic diagram shows the aqueous humor flows out through these, between the spaces of the beams that are lined by trabecular meshwork cells. And then the spaces get tighter and tighter until they come up against the wall of Schlemm's canal. And then the liquid has to go through by paracellular permeability and then enters the blood vessels to escape. Now, when something goes wrong here in primary open angle glaucoma, it's not really understood why it goes wrong. We see a pathology that's characterized by a loss of these trabecular meshwork cells, a collapse of the beams, and extracellular deposits. So it seems that there's some problem at the cellular level. And this is, we see in primary open angle glaucoma, but we also see it in the glucocorticoid-induced glaucoma. So the two seem to be very similar pathologically. Okay, so this was one of the genes that came up very high p-value. It's called GPR158. It's a G protein coupled receptor. And it had been completely unstudied. There was no literature on it. So we did a little analysis by bioinformatics to see